All right, we have been talking about the adaptive immune response, and we were looking in the last video specifically at the humoral immune response. And with the humoral immune response, we talked about the fact that the lymphocytes will make those clones, and the clones will become either plasma cells or memory cells. The plasma cells are going to secrete these proteins called antibodies. And that's what this video is going to be about. So antibodies are also called immunoglobulins or Igs, and they're capable of binding specifically with an antigen that's detected by B cells. They're going to be grouped into one of 5 classes or Ig classes. The basic overall structure of an antibody is usually a T or a Y shape. Antibody monomer is going to consist of 4 chains linked by disulfide bonds. You know what? Just know that it's Y or T shaped. This is basically what an antibody looks like. You can see it says heavy chain, light chain. Don't worry about all that. We're not going to get into any of that. This is another image of antibody structure. And then we're going to start talking about the 5 major classes, the five major Ig classes. So we have IgM, IgA, IgD, IgG, and IgE. IgM is a potent agglutinating agent. IgA is going to be found in mucus as well as other secretions, and what it does is it helps to prevent pathogen entry. This is just kind of looking at the structure. Don't worry about the actual structure of the antibody, and really just focus on what we're talking about in this video. IgD, attached to surface of B cells, functions as B cell receptor. IgG is going to account for most of the antibodies, 75 to 85% of antibodies in plasma. And this is from secondary and late primary responses, and this particular antibody can cross the placental barrier. Finally, we have IgE, and then you can think of IgE for allergy, and it's going to be active in some allergies and parasitic infections. And this is going to cause mast cells and basophils to release histamine. And we talked a little bit about histamine. It's a vasodilator. And remember, you take an antihistamine because your eyes get watery and things are itchy. And that's what histamine does. This is just looking at those 3 antibodies we just talked about. So, antibodies, what they're going to do is they inactivate and tag antigens. They do not destroy them. They form these antigen, antibody, or immune complexes. And we have defensive mechanisms used by antibodies listed down here. It's neutralization, agglutination, precipitation, and complement fixation. Neutralization is the simplest, but actually one of the most important defensive mechanisms. The antibodies block specific sites on the viruses or bacterial exotoxins. And what this is going to do is it's actually going to prevent antigens from binding to receptors on tissue cells. At that point, those antigen antibody complexes will undergo phagocytosis. Agglutination. So antibodies can bind same determinant on 2 different antigens at the same time. Remember, antigenic determinants are where the antibody binds. So antibodies can bind the same determinant on 2 different antigens at the same time. This is going to cause these antigen-antibody complex to become cross-linked into these large clumps. This process is called agglutination. An example of this is going to be clumping of mismatched blood cells. This is just looking at agglutination right here. We can see them attached to multiple red blood cells here, and then we form these kind of cross-linked lattice type clump things that are going to be phagocytized, like taken up by the immune system. Precipitation. So this is where soluble molecules, not cells, are going to be cross-linked into complexes. These complexes will then precipitate out of solution. And once it's precipitated, these complexes are easier for the phagocytes to engulf. The last one we have is complement fixation and activation. This is the main antibody defense against cellular antigens like bacteria and mismatched red blood cells. Don't need you to know anything else about this. This is just looking at all the different mechanisms. Neutralization, again, we're just blocking the sites. Agglutination, we're clumping them up. And then precipitation, this is where we're clumping soluble molecules. And then we have complement activation. So to kind of sum everything up, antigen antibody complexes do not destroy the antigen. It prepares them for destruction by innate defenses. That is all I have for this video, and this is going to be the last one I post. Everything else we're going to cover in class.